Okay, so this video is for biochemistry on the paper by Gonzalo in 2011, and today's date is the December 11th. And for background information on this paper, monooxygenases perform selective oxidations of organic substrates under environmentally small conditions. They're regarded as extremely valuable biocatalysts. Its applicability is often hampered by their dependence on nicotinamide cofactors. These cofactors are required to deliver electrons to the oxidizing cofactors such as HEM and P450 monooxygenases and flavin and flavoprotein monooxygenases. Light can be used to reduce cytochrome P450 and flavoprotein monooxygenases. By flavin cofactor redesign, several artificial flavoenzymes were created that function as peroxide-driven monooxygenases, capable of enantioselective sulfate oxidations. Flavoprotein monooxygenases represent a major group within the superfamily of monooxygenases. A key enzyme intermediate in the catalytic cycle is the so-called 4A hydroperoxyflavin, performed by reduction of the flavin, typically at the expense of NADPH. Most flavoprotein monooxygenases can form and stabilize such intermediates, but some are unable to do so, and it's known that peroxyflavins quickly decay when free in solution. In order to develop a flavoprotein monooxygenase that operates independent from NADPH, they focused on flavin cofactor redesign. While natural flavin cofactors are unstable in their peroxy form, N5-alkylated flavins can form stable 4A peroxyflavins upon reaction with hydrogen peroxide. Studies have shown that these flavins are powerful oxidizing catalysts. And in the study that they did, they selected riboflavin binding protein, which is RFBP, from chickens, which has no catalytic activity but has a capacity to bind the riboflavin. A concise selection of alkylated riboflavin and lumeflavin derivatives were prepared. APO-RFBP was prepared and reconstituted with flavin derivatives. Incorporation of the reduced forms of these flavins into RFBP was successful in all the cases. And from table, I decided to interpret table 1 from the paper. This is describing the table. After reconstituting APO-RFBP with these flavins and verification of tight bonding, the derived synthetic flavoproteins were tested for their ability to perform peroxide-driven anion-selective sulfoxidations. The created artificial flavoproteins were active as catalysts and even displayed an enantioselectivity. Using flavin 2 bound to the RFBP, all tested sulfides could be converted, yielding the R sulfoxides in excess. The use flavins 3 to 5 yielded similar yields and optical purities of sulfoxides, but within most cases in excess of the S enantiomers. When using flavin 4, a competent peroxide-driven monooxygenase is formed, which forms R sulfoxides. The results indicate that by increasing the size of alkyl moieties of the modified lumeflavins, and the anion selectivity is decreased. And the graphic R1 and R2 are different R groups for the amino acids, the column R1 and R2. The flavin column represents all the different flavins they use to test their ability to perform peroxide-driven enantioselective sulfoxidations. The last column represents each flavin's compatibility with R sulfoxides or the S enantiomers. The higher the percentage, the greater chance the flavin has fit with each sulfoxide.